Hello, my name is Carolyn and I am a part of the non-small cell lung cancer team, and this is our presentation of our research. Lung cancer accounts for 11.6% of all cancers, yet it accounts for 18.4% of all cancer deaths in the United States. The higher death rate is due to lung cancer commonly being detected in the later stages. To respond to these growing numbers, the search for biomarkers for early detection, diagnosis, and/or prognosis is a fast-growing necessary field. Lung cancer is split into two main subtypes, small cell lung cancer and non-small cell lung cancer. Our focus is non-small cell lung cancer, which has its own subtypes as well, large cell carcinoma, squamous cell carcinoma, and adenocarcinoma. Our paper outlines the current landscape and challenges in emerging circulating DNA and microRNA-based biomarkers from liquid biopsies for the prognosis of non-small cell lung cancer. Based on the current literature, circulating nucleic acid biomarkers appear to hold promise in indicating targeted treatment plans for non-small cell lung cancer patients presenting with specific genomic profiles. With the increasing number of emerging biomarkers, it is critical to analyze the clinical utility of these biomarkers in light of their prognostic value and clinical indications. The formal definition of a biomarker is a measurable indicator of the severity or presence of some disease state. Examples include pulse and blood pressure, blood lab tests, and tissue lab tests. Numerous biomarkers are discovered each year, but only a few biomarkers are currently used in clinics. The clinically acceptable biomarkers are confirmed via invasive tissue biopsies rather than non-invasive liquid biopsies. Tissue biopsies are invasive and require solid matter from the body, usually removed directly from the tumor or from bone marrow. It is more time-intensive and it is not as easily obtained as liquid biopsies. Liquid biopsy is an emerging, minimally invasive process that potentially addresses the pressing requirements in early diagnosis and in monitoring of the disease. In lung cancer patients, liquid biopsies could capture the molecular diversity of the disease. It is quick and easily obtained. Two categories of biomarkers presenting particular interest in multiple tumors, including lung cancer, are circulating tumor cells and circulating cell-free nucleic acids such as DNA and microRNA, which will be delved into later in this PowerPoint. ctDNA, which is circulating DNA, is detected in the blood by the shedding of the DNA of the tumor cells. It is very useful in finding mutations in the tumor cells and helps guide targeted therapies. The common way to detect ctDNA is through liquid biopsies. Now I'll go over the different ctDNA technology. First up, we have fluorescence in situ hybridization, or FISH. With this, DNA fluorophore labeled fragments bind to their complementary strands. This method is applicable for blood, cytology smears, and bone marrow, and can detect whole chromosome losses or gains, duplications, and deletions. Next is immunohistochemistry, or IHC, which is where antibodies adhere to antigenic determinants. This is used to assess proteins most commonly, but also nucleic acid expression. Next-generation sequencing, or NGS, is parallel sequencing of genomic segments. These segments are then compared to the human reference genome to see where it matches. Lastly is polymerase chain reaction, or PCR. For PCR, primers and nucleotides are added to replicate the specific genomic sequences. The three steps are denaturation, annealing, and extension of the nucleic acids. DNA can be further assessed using techniques such as gel electrophoresis. Now we're going to look at some of the ctDNAs that will be talked about in our paper. First up, we have epidermal growth factor receptor. EGFR mutations are found in up to 35% of adenocarcinoma patients. EGFR targeting tyrosine kinase inhibitors have been shown as a good form of treatment with good prognosis. Unfortunately, about 30% of patients develop resistance to these TKIs. Secondly is ERBB2. Approximately 1 to 5% of lung cancer patients have ERBB2. Some studies have shown that this percentage is about 1 to 2% specifically for non-small cell lung cancer. Results of one study demonstrated that 44% of patients with ERBB2 exhibited response to targeted therapies. Although the sample size was small, clinical indications and availability of targeted therapies for ERBB2 may warrant the use of these treatments, though the authors caution that genotypic differences in ERBB2 mutations may result in differential efficacy of targeted therapies. Trastuzumab, in conjunction with chemotherapy, has shown promise in clinical settings, although it has not been shown to penetrate the blood-brain barrier, rendering it less useful for metastasis to the brain and spinal cord. Other promising therapies include dicotomimab and neratinib. ALK mutations are most common among patients with a light or non-existent smoking history, adenocarcinoma histology, a younger age, or female gender, or those with wild-type EGFR and KRAS. EML4-ALK fusion variant 3 is the new target for a possible biomarker for higher risk cases. Presence of this specific protein causes acceleration of the disease, failure at early stages of treatment, and poor overall survival. Crizotinib plus immunohistochemistry is considered standard for detection and treatment of ALK. Multiple therapies are in the market, but fusion variants of ALK still need to be studied. Next up is KRAS. G12C is the most common mutation and is directly linked to smoking. KRAS G12C proved to be an important predictive biomarker compared to the other KRAS mutant types. There have been some attempts to treat KRAS patients with EGFR TKIs, but they have failed. Although there is no TKI in the market, KRAS G12C inhibitors are showing results in ongoing clinical trials. BRAF mutations are present in 3 to 5% of all non-small cell lung cancer patients. There are two types of BRAF mutations, non-V600E BRAF mutations, which are oncogenic in non-small cell lung cancer, and V600E BRAF mutations. The major problem is that targeted therapy for V600E BRAF has resistance to non-V600E BRAF, so there needs to be more studies done on it. PIK3CA regulates multiple tumor-relevant regulatory processes such as cell growth, survival, and cycle progression, and is present in 2 to 7% of all non-small cell lung cancer patients. It is especially pertinent in squamous cell carcinoma, but its role in prognosis lacks supporting evidence and is inconclusive. For ROS1, the mechanism of mutations is currently unknown. In general, ROS1 rearrangements occur in the absence of other known oncogenic driver mutations. miRNAs are a class of non-coding RNA that regulate gene expression and serve as chemical messengers to mediate cell-cell communication. The biosynthesis pathway of an miRNA is that it is transcribed from DNA sequences into primary miRNAs and processed into precursor miRNAs and finally into the mature miRNAs. They exhibit dynamic interactions with target genes, including inducing or inhibiting transcription and translation. miRNAs can be secreted into extracellular fluids and transported to target cells via exosomes or by binding to the proteins. And this video is a small clip that helps explain miRNAs.
This is a cell, the basic unit of all living organisms. Our body consists of different cell types, like skin, muscle, or bone cells. They have different structures and functions, but contain similar components, like the nucleus. The nucleus contains the DNA that encodes the instructions for cell development and function. This information is arranged into segments of DNA called genes. Almost every cell contains identical copies of DNA. But if there are the same instructions for all cells, why are there so many different types? The reason is gene silencing. Genetic information can be switched off. So, during development, a cell only reads instructions that are necessary for gaining the characteristic structures and functions. For gene silencing, a cell can use a microRNA. This powerful molecule is able to turn off genes by inactivating messenger RNAs, which are necessary for translating the genetic information into proteins. In this way, genes can be turned off. MicroRNAs participate in the regulation of the cell from its development to its death. Their dysregulation can have serious consequences for our body and can cause a range of diseases such as cancer.
miRNAs average 20 to 22 nucleotides in length. Extracellular miRNAs have been widely reported as potential biomarkers for a variety of diseases, and they also serve as signaling molecules. In fact, it is well established that these small RNAs recognize their mRNA targets mainly by the 2nd to the 8th nucleotide of their 5' end. Mismatches in the 3' sequence allow one miRNA to specifically bind to hundreds of different mRNAs, simultaneously regulating their expression. Now I will delve into some specific miRNAs used for prognosis in non-small cell lung cancer. First up is MIR155. Overexpression of this miRNA can be used as a characteristic in cancer. Expression is higher in those with recurrent cancer as opposed to newly diagnosed. Higher expression has a negative role in prognosis for squamous cell carcinomas, yet a positive role in adenocarcinomas, which is very interesting. High serum MIR21 level is associated with a low 3-year overall survival. Serum MIR21 levels following non-small cell lung cancer resection predicts tumor recurrence and might be linked to platinum-based chemotherapy sensitivity. The expression level of MIR125B predicts chemotherapy sensitivity as well. High expression is significantly correlated with poor survival. The MIR30 family includes 5 members: MIR30A, MIR30B, MIR30C, MIR30D, and MIR30E. For MIR30A, low plasma level is associated with high malignant potential, low E cadherin expression, which is a tumor suppressor, and high N cadherin expression, which is associated with poor cancer prognosis. For MIR30B and MIR30C, high plasma levels of both are associated with an inferior effect of erlotinib in EGFR-mutated non-small cell lung cancer patients. For MIR30C and MIR30A, high expression of these miRNAs and low expression of TM4SF1 correlated with more prolonged overall survival and progression-free survival in non-small cell lung cancer patients. MIR30E is considered oncogenic or tumor-suppressing, and overexpression of it improved lung cancer sensitivity to gefetinib both in vitro and in vivo. For LET7, low expression is associated with poor overall survival rate and prognosis. For MIR145, expression level in the tumor tissues was significantly lower than in normal tissues and was lower in chemotherapy-resistant patients. After 20 months, low MIR145 expression had a disease-free survival of about 50%, whereas high MIR145 expression had a disease-free survival of about 90%. On control non-cancerous cells that were transfected with MIR145, the cells proliferate at a lower rate. On the other hand, cancerous cells with MIR145 expression that was suppressed proliferated at a higher rate. These results show that MIR145 is a potential tumor suppressor. High expression level of MIR486 is associated with shorter overall survival. MIR486 suppresses invasion and migration abilities, preventing metastasis. MIR486 is an effective therapeutic agent for improving cisplatin sensitivity, reducing chemotherapy resistance. Unlike standard tissue biopsy options, ctDNA and miRNA are obtained via less invasive methods, usually via a simple blood test. However, the question remains whether ctDNA or miRNA will serve as viable standalone methods in the detection of actionable or prognostic biomarkers. Currently, testing incorporates both standard tissue and liquid biopsy methods. To determine the efficacy of liquid biopsy techniques, further research must be conducted to determine the concordance rate between liquid and solid biopsies. While liquid biopsies may provide additional benefit by circumventing the issue of intratumor heterogeneity within solid tissue biopsies, it is unclear whether circulating nucleic acid biomarkers exhibit differential release. In this case, the benefits of circulating nucleic acid biomarkers is tempered by confounding factors such as the amount of measurable ctDNA or miRNA. Based on the current literature, circulating nucleic acid biomarkers appear to hold promise in indicating targeted treatment plans for non-small cell lung cancer patients presenting specific genomic profiles. With the increasing number of emerging biomarkers, it is critical to analyze the clinical utility of these biomarkers in light of their prognostic value and clinical indications. Clinical actionability remains a significant factor in determining biomarker value. Current literature suggests that early detection of specific biomarkers can provide a remarkable clinical benefit. Ultimately, the prognostic significance of ctDNA and miRNA biomarkers in non-small cell lung cancer care has demonstrated that liquid biopsies and molecular diagnostic testing may yield profound increases in overall survival rates. Further testing must be conducted to analyze the significance of these biomarkers in larger cohorts and to determine the precise clinical benefit of their standardization. And here's the acknowledgments for everybody on the non-small cell lung cancer team. Thank you for listening.